So just to reorient everyone and make sure we all start on the same page, our problem statement was to develop a framework to derive an in vivo transcriptome point of departure for use in chemical risk assessment that will produce a human health protective point of departure based upon concerted molecular change. Now, I think this landscape has broadened a little bit. The term in vivo, while it is still the primary focus of the group, we have a bunch of people that joined, Jason being one of them, that works in, I guess you could argue that he works in vivo, he works in ecospecies, so this not rodents, but then there are others that do a tremendous amount of in vitro work. And so there's a sort of a collective framework that we'd obviously like to be able to adapt to all of those systems. So as you can imagine, this is a relatively complex problem. So we really, in some ways, we're fundamentally a framework development and evaluation group and a community of practice whose goal is to identify areas for improvement and provide guidance on the application of genomic dose response and risk assessment, although we are still very much focused on the human health protective point of departure. And a lot of this is, what's happened is, and I'll discuss this a little bit later, is this approach has evolved to such a degree that there's interest in regulatory agencies in the application of it. And so more and more people are going, hey, well, I'll try it. And then they try it and they come to me and they say, well, that didn't really work for me. What do you think I should do? Something's, there are just challenges in the evolution and codification of the approach. And so this group is very much involved in those sorts of discussions and sort of collectively providing a guidance on how to do that. And we actually started out at a very philosophical level, which I'll touch on in a minute. And so it's very much, shared experiences and then being able to offer a sort of a landscape characterization of the approach and then some guidance. So we have a bunch of great contributors, 7 from industry who have been outstanding, have been very actively involved, a bunch from the academic sector, and then, of course, we have a lot of interest in the nonprofit. And then I really have to say thank you tremendously. I also have to say thank you to Connie and Chrissy, because they have been amazing at supporting this group and keeping us organized. As you can imagine, when you're dealing with a broad landscape problem, trying to narrow the scope and getting into a collection of coherent ideas that you can formulate and put out to the world is a challenge. It's not a set collection of experiments that you run and write up. It's more of it's trying to wrap your hands around a very large challenge and a very large landscape. So we've done some proselytizing of our ideas in 2024.
Scott, for me, it's like 10 seconds delayed when the slide changes. I don't know about for others. Okay. Maybe just hit it before you're going to change.
I will slow down.
But we see it now. I see the SOT slide.
Okay. So there was an SOT workshop that was put together by a collection of people from HESI, a number of which are participants in the group that in the working group. The title of it was Application of Early Molecular Measurements to Develop Points of Departure for Risk Assessment, and it covered a pretty broad landscape, obviously the in vivo, but also some of the other areas that you see Josh Harrell there, who is obviously very much on the in vitro side, and then others that are involved in the eco and other technologies using a similar sort of dose response analysis, which you heard about a little bit earlier from the OASIS group. And then I hear, I wasn't there, but I hear that our symposia at EMGS was standing room only. And there's Cam up there giving a presentation on a slide you'll see farther down in our list here on the sort of the grander philosophy of our approach. And this was put together by Logan Everett and Cam. I believe that was supposed to be Jason O'Brien actually presenting, but he couldn't make it because of I believe sponsorship issues or payment. Basically, Health Canada decided, or Environment Canada at the last minute decided it was not appropriate for him to attend. So Cam stepped in and gave the talk. And again, I heard it was extremely well attended, although I was not there. In addition,
I'm sorry. It seems like a lot of other people are having it. Why don't I just take sharing? I'm sorry for the tech issues and to interrupt you.
Yeah.
But that way, I'm sorry, but you should be able to see it now.
Yeah. Okay. So next slide. So this was the talk from EMGS. There was also a poster presented at Eurotox by Karen Tillman, again offering up our sort of grander picture of the application of transcriptional points of departure in chemical testing. Next slide. So here's the slide that you may have seen Cam presenting in the slide that I probably should have waited to talk about. But this was the first paper that the group put out, and it was sort of the fundamental philosophy. And at the time, Cyril Pettit was helping nurture the group forward, and she did an outstanding job at the tail end of this, bringing together a diverse collection of thoughts with the help of a number of people in the group to lay down these 4 principles that undergird the sort of philosophical approach to transcriptional points of departure. And so principle 1 was transcriptomics is a reliable tool for detecting alterations in gene expression that result from endogenous or exogenous influences on a test organism. I think we generally agree upon that. Principle 2, alterations in gene expression are indicators of either adverse or adaptive biological responses to stressors in an organism. Again, I think that's probably acceptable. The community would largely accept that. Principle 3, they get a little bit more challenging as we go up, but in general, I think we have agreement. Principle 3, transcriptomics can be employed to establish a BMD-based point of departure from short-term in vivo studies at a dose level below which a concerted molecular change is not expected. And this is the idea that molecular has to occur before phenotypic. And I think that's generally accepted. I think we're coming across, as with all things toxicology, we come across exceptions, but for the most part, that is true. And then the use of a transcriptomic point of departure is principle 4 here. The use of a transcriptomic point of departure set at the concerted molecular change level will provide strong protection for use in risk assessment. And again, that will, as we go on in time and we evaluate more chemicals, that will accumulate the evidence for that. But currently, as it stands right now, that evidence appears to be pretty solid. Next. Oh, and if you want to read the paper, there's a link there with the QR code in the lower right. So once we got the philosophy down, a lot of what we began discussing in the group were the challenges with different data types and modeling and how we approach going all the way from raw data to quantifying uncertainty in T-pods and determining T-pods. And there was a lot of work prior to this group taking it on. There was a lot of work. Carol Yalk produced a tremendous amount of work in this space, amongst others, in evaluating how you run pipelines and identify points of departure. And so but every time, people would run it with in vitro data, people would run it with ecospecies data, cell lysate data, FFPE data. And what we were finding is there needed to be refinement based upon different variables and that were part of the study. And so we went through as a group and formulated this manuscript based upon this outline here on the upper part in blue. We reviewed the current state of science regarding the bioinformatics workflow associated with the T-pod derivation, and we identified known variation, sources of uncertainty, data gaps, and recommended potential ways to address them in the paper. And then we also stepped a little bit outside the boundaries of what we're currently doing and talked a little bit about how there's potential evolutionary steps in the analysis, looking at things like modeling latent variables, integrated variants, gene set scores, things along those lines. So we imagine this approach is going to keep evolving as the science keeps evolving. And as a spinoff of that, we formulated a plan for a workshop to first, we're going to invite leaders in the field to investigate the most pressing issues that were identified in the paper and go through and identify a collection of existing data sets to use as case studies while at the meeting to evaluate certain endpoints or plan out evaluations both before, during, and after the workshop to get some more solid answers on on a number of the questions that occur along the analytical pipeline. And then, of course, put out additional publications and workshops related to the case studies that we've identified and then evaluated. So we'll point out just one last thing. I didn't mean to skip over this. The paper is in press right now in ToxSci, and the citation is right there in the middle of the slide. Next. So right now we're in the process of preparing for this workshop because this is my first bullet point there. This is fundamentally a complex optimization problem. So there are a number of different variables, and they have a tendency to interact. And so depending upon how many dose levels you apply, how many dose levels you use, it will determine whether or not it's appropriate to use certain criteria for filtering data and things along those lines. And so there's this basically this cascade of questions that we are trying to create and evaluate whether or not we have existing data in the literature that can answer that, what metrics we would use to answer that question about that parameter in the analysis pipeline, or if we don't, whether we have an expert opinion that's available that we would trust. And again, going back to that, if we don't, what data do we need to produce? So just to give you an idea of some of these variables, there's context of use, and I've talked about this before, right? I work in vivo with purified RNA. I know the ETAP people, which I'm going to touch on the ETAP in a minute here, the EPA product, also do the same thing. But then we also have in vitro groups, and I've been involved in a number at NTP where they use cell lysate. And EPA has apparently tried to use BMD Express on cell lysate with their protocols, and they've run into some very significant challenges because of the increased noise in the data. And then there's the question of, whole organism versus isolated tissues or individual cells, things along those lines. And so we have this purified RNA, like I noted before, or lysate, which you get from in vitro, or alternatively, what I didn't list on here, a number of groups started off using FFPE, which is very much akin to, I don't know if it's akin to a lysate, but it is definitely different than purified RNA. So the parameter settings are going to be a little bit different because there's going to be more noise in the system. And then there's all sorts of questions along the analysis pipeline. And, being a toxicologist, the first thing you talk about is dose selection. And that is a pretty huge thing if you want to detect signal. A number of the people that are in the working group have different dose selection criteria than others do. And so it can be challenging sometimes to pick up signal. Number of dose groups, depending upon the chemical that you're dealing with. It could be as few as 5, as many as 10. Number of replicates, that plays into how you're doing the modeling. And then what tissues you're going to cover. Now, I don't think we're going to cover some of this stuff, but these are considered in the workshop. We're probably mostly going to stick with the bioinformatic pipeline, but these all play into the bioinformatic pipeline selections. And then you have the technology selection, data QC, and filtering. So one of the things is if you have 0 counts for a given gene in a sample set, unless you use a censored approach, it's probably not appropriate to be modeling the data with a modeling where you have, say, a couple of zeros in your data, or even 1 0 in your data for a gene. If that's there, you basically have to run a censored data analysis, which is a quite sophisticated analysis. So, the question is, do you leave those in and suffer the consequence of modeling data that has 0 count values and things along those lines? Then there's the response detection method. So once you've got the data into the analysis tool, how do you identify differentially expressed genes that you would subsequently like to model? There's trend tests, there's curve fits, there's ANOVAs, activity thresholds, meaning or effect size thresholds, which is a fold change value. And then, okay, so now you've filtered down, you got a bunch of genes that you believe are dose responsive. How do you fit a dose response curve to them? And this is an area of rapid evolution, primarily because of the motivations in the guideline data space, where there are real significant mathematical challenges because the number of dose levels are smaller. But here we tend to deal with more. But still, there's this question of what's the best way of fitting a line to data. And then BMR type. The general philosophy with continuous data is a standard deviation-based benchmark response, typically a 1 standard deviation or 1.349 standard deviation. But if you look at the literature and you're looking for the most repeatable by rank BMD from experiment to experiment, if you ran the same chemical, you probably want to use a relative deviation, like a fold change. Kind of knew that from MAQC, but we're rediscovering it. I think we're going to rediscover it here. And then how do you determine a point of departure? And the uncertainty around it. We have distribution-based methods. We have gene set-based methods. What's most appropriate? And then finally, once you've created your pipeline, how do you characterize false discovery? And then there's other variables that play into this. The genome annotation. So Jason has brought it to our attention oftentimes that the annotation of the genomes that he's dealing with are not anything like a human or even a rodent. And so sometimes maybe the gene set approach isn't the best way to be analyzing that data. Then there's a question about how do you model the data to get the greatest repeatability and reliability of the results? I mean, one of the things that I've been struggling with is in my work, and I think other people have too, is there's a range of basically false discovery of response. And then there's a range just above that appears to be signal above background, but if you ran the experiment again, it disappears. It completely changes. And then there's a region right above that. That's what we consider typically repeatable. And that's generally at least 1.5 to 2-fold change, right? There's other variables that go into that, z-scores, things along those lines. But so what do we do with that intermediate range? And how do we manage that? Because if all your data is in that intermediate range, what do you say about that study? So there's that question. And then finally, obviously the biggest thing is maintaining, once we develop this pipeline, the original goal was the agreement with the apical points of departure. As long as we can maintain that agreement we have an existing pipeline that does that, do we maintain that with any sort of new pipeline that we come up with? And we're going to play with other things along those lines, but with each one of these that you're looking through here as variables and choices that you have to make, what's the metric for parameter selection? What am I going to do? And what am I going to do to select between relative deviation versus standard deviation for a BMR? And so those sorts of questions need to be answered also. And then finally, one of the great things, and EPA's helped contribute to this with their ETAP project, is identification of data that can be used to empirically address the questions and where expert opinion is, of course, appropriate on this. So there are a number, as you can see here. Again, I've tried to, as we're getting down into the details, you realize there's a pretty massive complexity to this process, and a number of these variables can interact. So it's why we're slowly working through this, trying to identify the most optimal set of variables and parameters that will give us data that is both reliable, repeatable, and agrees with an apical point of departure. Next. So just to emphasize, we are holding this workshop in Durham at the US EPA the 24th through the 26th. There will be up to 50 government, academia, and industry NGOs and CROs, consultants from across the globe that will meet there. And hopefully we will get through all the complexity that I just outlined. I have high hopes. And we will publish case studies supporting the parameter selection and the various decisions that the conclusions, I would say, that we've come to as a group, and then identify research gaps and potential need for more experimental work. Next. So one last thing I just wanted to highlight here that I think is, at some level, a credit to a lot of the people that are participants in this group. And this group formed approximately 4 or 5 years ago now. This ETAP product, what I'm going to tell you about, came out in 2023. And before this group was formed, there were a number of us that have been working in this space. But collectively coming together, I think it's helped nurture an understanding of the application of the approach, and that has helped move things like this ETAP forward. So let's just talk about the ETAP a little bit here. Many of you are familiar with EPA environmental assessments. There is a number of layers. There's IRIS assessments where there's a tremendous amount of data. and a lot of time to develop them. PPR-TV is going down the list there, human health toxicity assessments. And now we have the ETAP, which is the EPA transcriptional assessment product. And they actually issued their first report in parallel with the review of the ETAP approach. And so I was actually a participant in this. I helped in the development of the ETAP. I was one of the contributors. I believe Cam was on one of the review boards. I believe Cam was on the value of information assessment component of this, if I remember correctly. And so what was done was EPA said, hey, we're gonna create this new report, and it's going to be based upon in vivo transcriptomics. The NTP had put something out in 2018. It was generally well accepted. A number of people in the group here participated in that process. And EPA saw an opportunity with that acceptance and all the background work that a lot of people had done to formulate a product that they could use for a rapid response, or shouldn't say rapid, but it is definitely quicker than a guideline characterization, particularly a 2-year cancer bioassay, to identify a point of departure and then go through the process of setting health-based guidance values. And so on July 11th through the 12th in 2023, there was a review of the actual ETAP product that they put forward. And then subsequently, later in July, there was a value of information characterization, which I think really went a long way in driving support for the ETAP. And this value of information, basically, it was carried out primarily by Dan Krusky. It basically evaluated the value of the information you get from a 2-year cancer bioassay, which takes on average 8 years to complete, at least in the context of the government. And then the ETAP, which is basically a 5-day in vivo genomic dose response study evaluating genomic dose response in 10 to 12 tissues in both male and female rats. And the conclusion was, in general, based upon primarily the speed by which it could be done, that the ETAP had greater value than the cancer bioassay. Now, of course, there are exceptions to that, but that was the general conclusion. And so combined, they've got a set of protocols and a clear assessment of value. And so subsequently went through review, and it was generally well accepted. And next slide. And as a part of this, and I should have mentioned this earlier, they did an extensive review of a lot of the existing data in the literature. And this review was, looked at a tremendous number. It was some of Rusty Thomas's old studies, Carol's studies. I believe some of Cam's studies were in there. My studies. There was a number of people that did FFPE studies, and that were all basically taken in and reviewed, and was a major supporting component of pushing the ETAP through and getting people to believe the value of this product. And one thing I would point out is that POD, again, I noticed it before, but the POD workgroup members were highly cited for their contributions to the literature that helped define and build the confidence in the approach. So I think it really speaks volumes to the value that we can continue to contribute as a group. Thank you. Thanks, Carol. Next slide. Carol's trying to clap me off the stage, I'm pretty sure. Okay, that's good. That's right. Stop. So I want to go home. So just so everyone understands, there's a standard methods document for the ETAP. It's more specific normal guidance. There's a method subject to peer review and public comment, and it was the whole thing was the protocol itself was subject to peer review and public comment. And the thing that they tried to emphasize, so it wasn't trying to compete with anything with data, is they focused only on data poor chemicals. Now, the irony of that is there are people, to my understanding, that really want this data in parallel with their guideline in vivo studies because they're deeply intrigued by the prospect of using it, but they're trying to socialize it and get it, become more comfortable with it. And then to make things quick, they have a standard ETAP template, and it parallels very much what the NTP was doing, which is potency only. And in ETAP world, I've said this a couple times, my running joke is First rule of ETAP Club is we don't talk about what happens above the point of departure. So it's just the point of departure. We're not talking about any biology at this point. We will, I imagine being scientists, we're eventually going to get into that issue of what is happening once you start activating genes. And then what's great about it is, again, the target time is 9 months from nomination to product. Pardon me, from initiation to release of the actual product. And so I think there's real excitement about this prospect. There's real excitement too about the prospect of eventually doing this in vitro. I think it's going to take some time to adapt to find some acceptance of that. I think what's been happening of late from my communication with people is they like the fact that they have parallel metrics that they are comfortable with, that you can pull from the in vivo studies like organ weights and clinical chemistries and hematologies, that they can pair up with the gene expression so they can get a sense of where the old data says they have a point of departure and where the new data says they have a point of departure. Next slide. Yeah. And so the goal of the POD workshop, is to help inform and refine those approaches that are used in the ETAP, and hopefully expand the applicability domain of the approach. Next slide. And with that, I have to say thank you to the group. It is I was listening to the last presentation, and there's like tremendous clarity of thought as to what experiments are being done and what analysis are being done, and we are framework building and grander philosophical issues are often more challenging, but it's about bringing people along and filling space with knowledge that makes people comfortable in accepting new ideas, and I think that's a really valuable aspect of what we do.
Thanks, Scott. Really well said. Maybe we can ask if Cam has anything to add, and then can open up for questions if Cam's still on.
I am still here. Thanks, Scott. That was really a wonderful presentation. Nothing to add. I want to thank Scott and everybody else in the working group for everything that they've done the last 4 or 5 years. I'm really excited. I will say one thing. I'm really excited for the EPA to use this. It's the first regulatory agency that has taken the leap to actually use this in a regulatory decision-making context. And I'm hopeful that other agencies can follow.
Thanks, Cam. Oh, I see Pierre has a question. Go ahead, Pierre.
Hey, Scott, great talk. In terms of the bioinformatics pipeline that you laid out, and you talked about some of the challenges with determining what's real expression level detection, noise levels, and limit of detection, things like that, effect size. And a lot of that may be related to sample size, right? You always have that challenge of where do you put your valuable resources? Is it in more sample size, or is it in more design of the particular experiment in terms of doses and points and things like that? So with your effort and the team's effort, what kind of communications are you having with the science size or the bench size in terms of wrestling those questions about what sample size is? And is there any effort to kind of standardize what that would be for point of departure type of experiments and studies?
There is. I mean, one thing I will point out is we've been working on synthetic data generation for a while now to address this question of power. And because obviously we can't run 500 animals per dose group. The point of this is to use less animals, right? And so what we've been able to do in collaboration with Sciome and Deepak Mav in particular is to come up with methodologies that retain intra-sample covariance and generate additional samples. So if you have 5 samples, what you can do is upload an entire dose response, so 5 animals per dose level, and then expand on that. So I can go from 5 to 500 if I want to, through a process of resampling that data, looking at the covariant, retaining the covariant behavior of the genes in the data set, and being able to generate samples and ask that very question that you're bringing up. And we also have paired replicate studies where we've done independent studies. And I wish I could say that there was going to be one answer, and there may be a convergence on this. I don't know, but there's going to be a practical compromise that relates to the fact that we want to keep these studies relatively small and moving quickly. But the variables that play into it are the different forms of data that we're pulling in. Now that we've, the group is somewhat extended beyond the in vivo purified RNA, and we're dealing with more noisy samples, those variables are obviously going to change, and those parameters are obviously going to change. One thing we can do is evaluate it empirically with what data we have, and that's what we've been doing. Fred Parham, I believe, is on the call, has been evaluating a set of repeat data at various parameter levels, trying to identify where we're getting at least consistency of results and repeatability. And we're actually building machine learning models based upon model parameters, given, of course, the power that you've pointed out, to determine what's a repeatable response and what's a non-repeatable response that will hopefully be built into the software that we made available. So, I know it's sort of a dance around the answer you're looking for, but I think the answer is it's going to depend upon the scenario that we're dealing with.
It would be great if I would see you dancing. Thanks, Scott.
Sure.
Thanks, Pierre. Thanks, Scott. There was a question from Neil in the chat asking about, with the advent of single cell and spatial transcriptomics, how do you feel, or what do you feel are the implications of these technologies on hazard ID and TPODs?
Actually, of late, I've been my light is hang up, and this is completely independent of the it's a follow on. It's probably a new work group for eSTAR, and that is where instead of saying we're not going to talk about what happens above the point of departure, we're going to start talking about it. And the single cell, I think, has actually been really informative, because we've started off looking at overt markers of toxicity, and what they are really just macrophages invading the tissue. So what happens is a bunch of these genes like GPNMB, which I know the Merck people, if they're still on here, know exactly what I'm talking about. All those genes that they've identified as pan tissue markers are basically markers of angry macrophages invading tissues. And so that's one component where I see single cell helping inform us. The other one is on the I don't know if Diane's still on here, but we had to talk about HNF4 downregulation and general stress. So most tissues, when you really stress them, they lose differentiation because they go into a proliferative state. So those biomarkers of hepatocytes or those highly expressed genes start to fade as you stress cells. And you can see this with chemicals like furan, that just scorch the earth of the liver, right? And it just the hepatocytes start to differentiate, the cells start to divide, and that's what happens. Now, on the quantitative point of departure side, the biggest challenge, I think, is cell type switching, because you're basically trying to, when you're doing single cell, and you say I did a dose response single cell. I know Tim Zacharuski's on here, but his former postdoc, whose name is, sadly, and I'm so embarrassed by this, is escaping me. Someone he did TCDD evaluations. I believe it was in mice looking at single cell. And one of the biggest challenge. What's that?
Rance.
Thank you. Rance, I apologize. Tell Rance I apologize. I love Rance. The biggest issue was being able to make sure you can keep the responding cells within a group, because you have to be able to group things. So if you're looking at responses in hepatocytes and things start changing, do you lose hepatocytes? Do you lose hepatocyte gene expression that is supposed to be influenced? And that's actually an hepatocyte responding, but it's changed so much transcriptionally to itself that it shifts to another region. You put it in a different group. So you lose that information. And so if we can figure out how to retain that, particularly in the higher dose levels where you are seeing a pretty significant shift, I think that could be really, really valuable. And I think I talked to Rance probably about 2 years ago now on that very issue, and I haven't followed up with him, but I think that's a real possibility. And it could be what's really neat about it is you can actually take the genomic dose response data now, and I've talked to Arun Pandiri about this, who's one of our pathologists, and you can actually take the single cell data for, say, hepatocytes, and if you use furan and do dose response, you can see the hepatocytes start to drop off, and you can see the inflammatory cells, like the genes associated with the inflammatory cells, start going up. So you can almost see the pathology in the dose response, which is kind of neat. But that's not exactly using single cell. It's actually post single cell. So I now have information on what's a marker for a cell, and you can trace those cell markers in the bulk tissue. So another variable there. Again, probably dancing around the answer to your question, but it is there's lots of potential applications to it.
Thanks, Scott. And I think that emphasizes the continued work. And I think you've mentioned this in your talk, figuring out going beyond. I know for the pharma side, we have some great pharma experts that dial in and kind of watching the space, but how to make it relevant when we do expect a change, right? How we can't just say, for industrial chemicals and pesticides, we're a little bit lucky.
Yes. Well, that was DJ. I mean, that was actually what brought on the conversation about having a farm representative on the in the group with Deidre. But obviously we have that potentially in our point of departure working group. But it is an issue because they will they have to know what's happening above the point of departure. Is it bad? Is it tolerable? Is it not tolerable? Is it reversible? Those sort of fun things.
Good. I don't see any more questions. I think, Scott, you did a really excellent job. I think I will have a plug that, that workshop is, kind of invite only, but if there are people interested in it and have the relevant expertise, we're definitely looking for that to make sure, it's wide reaching and we can work on those case studies and have people who can roll up their sleeves and help. So I think if you're interested in that, please let us know because that'll come up quick next year. Anything else, Scott, from you for TPODs?
I could talk for hours on this if you'd like, but I'm pretty sure people want to go home.